Hey guys, welcome back. So I'm going to talk about two things today. My first lecture, which is this video right here, we're going to go through regulation. So how we control how much protein we have, because we don't want to be making too much protein, we don't want to be making too little protein. How do we control it? We know that DNA makes the protein, but how does DNA control how much protein to make? And that's what we're going to be talking about today. The other thing we're going to be talking about today in my other lecture is going to be mutations, which is kind of a lot more interesting, I find, than gene control. But nevertheless, we're going to need to know both topics. Now, once we're done this, we only have one topic left, which is about DNA technology, and about cloning, and about DNA fingerprinting. So we're almost done, and just a reminder that we do have our unit test on Tuesday next week. Okay. So without further ado, let's do the regulation. Okay. So this is the big thing we're going to be talking about today, is controlling the amount of protein produced. Okay, we want to make sure that we can produce it in proper amounts. So first thing, okay, and very important to keep in mind, most genes are not active all the time. They must be turned on or off periodically throughout life of the organism. Okay, if you think of yourself, so right now you guys are mostly in grade 12, you're finishing your puberty. Well, during puberty, you had genes that weren't normally turned on around 11, 12, 13, depending on how old you were when you first started puberty. And then those genes remain on, and they remain on all the way until you're done puberty, which would be right around now they're starting to turn off. Okay? So it's important to remember that genes are not stagnant, okay? That they turn on and they turn off. And not only can they turn on or off, they can turn on to certain percentages. Like I can turn on 70%, or I can turn on 100%. Okay? And same with being able to downregulate it. So if I was at 100% and I wanted to produce a little bit less protein, I could bring it down to 70% or 60%. Okay? Now, certain genes that are always on, okay, and their levels are controlled, that are always, you're always on, are called constitutive genes or housekeeping genes. Okay. Oh, just to take a second here. Okay, sorry to interrupt there. But thank you to Megan, who sent me an email. I made a mistake when I was talking about transcription, saying that the mRNA is read from 3' to 5'. It's not. It's read from 5' to 3'. Okay, I just wanted to make that mistake. It was in my transcription lecture, okay, that I made an error when I was talking. So sorry for that little caveat there, but I just wanted to remember to bring that up. Okay. So first we're going to talk about gene regulation in prokaryotes. And most of the thing I'm going to talk about is prokaryotic regulation. Now, in my slideshow, I go through eukaryotic regulation, but that was more for my in-class class. Okay, so you guys aren't going to ask to know too much about eukaryotic regulation. Most of these can be prokaryotic. So just a reminder of prokaryotic, okay, our classic one is a bacteria. Doesn't have a nucleus, very simple genetics. Okay, so in prokaryotics, we can control the level of protein we're going to make 3 ways. Okay? So just to remember that protein is made from DNA to mRNA and then to protein. Well, we can control it at 3 different levels. The first level, we can say, well, let's not even make the mRNA in the first place. Okay? And that seems to be the most obvious way to not make protein, is to not allow DNA to make the mRNA. Okay? And that's our first level of control. Okay? This is also the most common level of control, coincidentally, because it's the most obvious level. Just don't make the mRNA in the first place. Okay? The other way is, what if we already have made the mRNA, so we already have the mRNA, but we don't want to make the protein? Well, the next level we can do is at the translation. So we could either destroy the mRNA or just not allow the ribosome to read the mRNA, or we could not cap and tail the mRNA. It's just preventing it from being actually read by the ribosome. And the last one was, well, what if you already made the protein and then you realize you don't need it? So you've already made it, it's inside your cell, and then your body sends you a signal that says, we don't need that protein, we've got lots of it. Well, that's called post-translation, because you've already made it, but now you don't want it. So we can still say eliminate the protein. We can destroy it inside the cell and then never have it released. Now that would be a huge waste. Okay, we wouldn't want to do it that way very often, but sometimes that just may occur where you didn't get that signal to not make the protein, it's too late, and you've already made it. Okay, so let's go through these a little bit more in depth. So in prokaryotes, the E. coli has been extensively studied. From these studies, we've noticed that prokaryotes cluster some genes together. Now this is in prokaryotes, not eukaryotes. So in prokaryotics, certain genes are always transcribed together. So it'd be like gene A, B, and C. Whenever A is transcribed, B and C automatically are too. They all go together. They're kind of grouped as one. Okay? And when this happens, okay, they're called operons. An operon is a term to include several genes that are all transcribed together. So with an operon, just one promoter binds, and once that one promoter binds, and this is where mRNA polymerase is going to bind, it controls the activation of all genes on that operon. So we have one promoter, and then all genes get transcribed based on that one operon. So therefore, in prokaryotics, one promoter activates a number of genes. Okay, so let's take a look at this. And I know this looks a little bit confusing, but let me go through it. So first, we're going to talk about the coding region. So this is where the genes are. Okay, so we've got 3 genes. We've got Z, Y, and A, and these are 3 separate genes. They're going to produce 3 separate proteins. Okay? Now, all of these proteins are required in order to digest lactose. Okay? Remember from our first unit, lactose is a sugar, and we want to break down the sugar. Well, breaking down a sugar is very complicated. We need more than just one thing. We need 3. We need Z, which is going to make a protein, Y, which is going to make a protein, and A, which is going to make a protein. Okay. Now, that's our coding region. Now, we also have a region upstream of it called a regulatory region. And a regulatory region is going to have 3 little areas. It's going to have an area for an operator, it's going to have an area for a promoter, and it's going to have an area, what we call for a cap. Okay? So the key thing is that all 3 genes are under the control of this one promoter. Okay? This thing controls the activation of all 3 genes. So now here's a better picture of it. Okay, so it's the same kind of picture. So here's our promoter region right here. Okay, here's our promoter region. And then here's our first gene, and there'd be a gene and gene and gene. So this is the lac operon. Okay, so the operon is the idea that we have several genes in a row, and this is the one that produces a protein that degrades lactose, and that's why they call it the lac operon. Okay? So it's got the coding region and a regulatory region. The coding region is simply the DNA sequence required to make the mRNA for the 3 genes. The regulatory sequence, pictured below, contains the promoter. So we got the promoter, and that's kind of all 3 things. Okay? An operator is right at the front. We're going to learn what these things do. And then a cat binding site, which is right back here. Okay? Now let's go through what each of these things do in the regulatory region. We already know what happens with these. These are going to make an mRNA and make a protein. But we're going to kind of focus here on regulation of genes, and that's why we're focusing on the regulatory region. Okay, the regulatory region contains the promoter, an operator, and a cap binding site. So this is the key things we need to know. The operator is an area that a certain protein, called the repressor protein, can bind to and prevent transcription. Okay? So if we have too much of these proteins being made, okay, so say we have too much of these proteins being made, we don't have any lactose that needs to be digested, therefore we don't need any of these proteins to digest lactose, so we need to shut off this. So how we shut this off is we have a repressor protein come and bind right to the operator. Okay? So then when RNA polymerase goes to the promoter and tries to get across, it gets stuck, it gets blocked by the operator. Okay? So the operator is kind of the stop button with it. Okay? And that's the point of the operator. Now if we remove the operator, okay, we're going to start making protein, just like irregular amounts of protein. Now sometimes we may need a ton of protein. What if the regular amounts of protein we're making by removing this operator isn't enough? Well, we kind of have like a boost. And a boost, it comes from the cap binding site. This is an area that a certain protein, okay, can bind to and ramp up transcription, making more protein. So we're going to have this CAMP, basically, molecule come in and bind to the cap area, and that basically ramps up transcription and makes more protein. So let's go through 3 scenarios. Now I want you to eliminate this middle scenario, okay, because it doesn't really make any sense. But the first one, if we have very low lactose, then we don't need this lac operon being active. We don't need to make any of these proteins. So we want to deactivate it. So if we want to make sure we're not making any protein because we don't need any because lactose levels are low, then we put on an operator. So there's my operator sitting there. RNA polymerase can't get down to these genes, and we get no transcription. If we move down here, and say we get a high levels of lactose, you drink a glass of milk, okay, or not me, but a bacteria drinks a glass of milk. I know it's ridiculous, but for this scenario. So they have high lactose levels. Well, the operator's got to come off. Right? And if the operator comes off, we're going to start making proteins, but not a ton of proteins. And we need a ton of protein because we have high levels of lactose. Therefore, we're going to have our cap come in and bind there. And once our cap is bind there, then we've got lots of promotion, and we're going to make a ton of these proteins. This middle one right here, okay, it should say medium levels of lactose here. Because with medium levels of lactose, you're going to want the operator to come off, and it did, but nothing binded to the cap site. Because we don't need extra transcription, we just need regular amounts. Okay? And that's controlling the lac operon. Now there's one more operon that we want to talk about. It's called the tript operon. And the point of the tript operon is to make tryptophan. And tryptophan is an amino acid that we use to make protein. Okay? So the tript operon is creates the amino acid tryptophan. Now, this is the thing why we want to talk about this operon. This operon is always active and only turns off when high levels of tryptophan are detected. So it's very different than our lac operon that has an operator and a cap binding site. With our tript operon, basically what happens is high level of tryptophan come in and basically stop this transcription from happening. It's like tryptophan almost acts as its own operator by coming in and binding there. Right? Because excess tryptophan will bind to a repressor protein. That's kind of my repressor protein right there, which will in turn bind to the operator, which in turn prevents transcription. Okay? So it's a little bit different than our lac operon, but the big thing here is that tryptophan is always on, and that tryptophan will bind to a repressor protein, which will bind to the operator, which then prevents further transcription. Okay. So the only thing I want you to know about eukaryotic gene expression, I'm not going to go through this in great detail because they don't in your content, and I won't ask you about it in great detail, is that you have one more level of control. Okay? When we talk about eukaryotic control, we have all the controls that we just talked about in terms of not making the protein, not allowing the mRNA, but we also have this pre-transcription control, and that's the idea that we never even let the DNA unwind. Right? So if we don't want to make protein, not only can we not make the mRNA, we can prevent the DNA from even unwinding in the first place. And if DNA never unwinds, it can never make its mRNA. And that's the pre-transcription control that eukaryotics have that prokaryotics don't have. Okay? So that's it for the lecture on gene regulation. Okay? If you have any questions or concerns, send me an email. Make sure you do your portfolio section in the content section, and I'll be uploading this right there.